Hello, my name is Sam Cox.
My name is Tristan Packard.
And my name is Matt Brown.
We were part of the BYU Phage Hunters program, and as a part of that program, we investigated tail assembly chaperone proteins of the Microbacterium foliorum cluster EE phages. In this process, we discovered a phage which we named BRE160 in our group, and by various methods after sequencing its DNA, we examined an anomaly that we found at the gene 10, 11, 12 area of its genome. Here are the normal tail assembly chaperone proteins, and we thought that gene 11 was demonstrating some strange behavior. So we used a variety of methods to try to investigate this further and came to some strong conclusions.
One of the tools that we're able to use was the Famirator map. And what we did was we used the BRE160 genome and compared it to other genomes of cluster E phages. What we found was that there was 4 tail assembly chaperone genes in our phage BRE160, and most other cluster E phages only had 3. And so what we did was we looked into this more and we found that we did have one more gene than other phages. And what we found was that it overlapped one of our genes. And so there's actually only other phage that had 4 genes, and that phage name was Azizum. So what we did is we put a Famirator map on our poster, and you can see and you can compare the other phages that we found.
So another method that we use to analyze the existence of gene 11 is using the iTaser software from Zhang Labs with the University of Michigan. Now, this software is a predictive modeler of the potential secondary structure of the proteins that would be coded for by these genes. So if we look at these pictures here in the middle, the first and the last picture show gene 10 and 12, which are common among all the cluster EE phages, this 10 and 12 protein. And looking at their pictures, you can see that they're very organized with a high degree of secondary structure and with tight alpha helices. Now, if we compare that to gene 11, which is shown from 3 different angles, gene 11 is very disorganized and it is not a strong structure. Now, the iTaser software also gives us what is called a C-score, which is just a confidence value of how predictive this structure really is for the true protein. For genes 10 and 11, 10 and 12, excuse me, it returned a C-score of 4.81. And that's pretty high. It's a fairly high confidence level. But there was no C-score for gene 11, which sheds doubt on the total existence of this gene at all in the genome.
In conclusion, we really felt strongly that gene 11 is not a viable protein, at least on its own. We would need to do additional wet lab research in order to find out if gene 11 is doing anything at all. And we think that if it is, the most likely candidates are either some sort of conjunctive activity with genes 10 or 12 or both. But this really could be just the manifestation of gene 11, the result of transposon activity. Thank you for your time.